One of the emerging frontiers in the science of aging and obesity is the role of cellular senescence, where cells stop dividing but resist death, becoming metabolically dysfunctional and pro-inflammatory. These senescent cells accumulate with age in various tissues, including fat, liver, and muscle, where they disrupt local metabolism, secrete harmful cytokines, the so-called senescence-associated secretory phenotype, or SESP, and promote insulin resistance. The clearance of senescent cells has been shown in animal models to restore tissue function, reduce inflammation, and improve metabolic parameters. While senolytics, drugs that specifically kill senescent cells, are still under development, there is growing evidence that NMN, through its NAD+ boosting properties, may enhance autophagy-based mechanisms for the natural removal of these harmful cells. This positions NMN not only as a metabolic enhancer, but also as a possible modulator of tissue rejuvenation through senescent cell clearance. Autophagy plays a vital role in maintaining cellular health by degrading damaged organelles and proteins, but it also contributes to the surveillance and removal of senescent cells. In youth, cells with DNA damage or mitochondrial dysfunction can either self-repair or undergo programmed cell death via apoptosis or autophagy-mediated clearance. However, with aging and NAD+ depletion, these quality control processes decline. As a result, damaged cells increasingly escape clearance and linger in tissues, secreting SASP factors like IL-6, TNFA, and MCP, one that create a pro-inflammatory microenvironment. This chronic inflammation not only accelerates tissue aging but also promotes adipocyte dysfunction, hepatic steatosis, and skeletal muscle insulin resistance, contributing to the progression of obesity in later life. NMN intervenes at a foundational level by replenishing NAD+, the essential coenzyme for sirtuin activity and mitochondrial health. By doing so, it restores the energy-dependent signaling required for proper autophagic function. Specifically, NMN reactivates SIRT1 and SIRT3, which play key roles in stress resistance and mitochondrial renewal, and indirectly regulates mTOR and AMPK pathways that govern autophagy initiation. This leads to enhanced lysosomal function, autophagosome formation, and degradation of damaged cellular components, including elements of the senescent machinery. In aging models, NMN supplementation has been shown to increase autophagic flux in multiple tissues and reduce the presence of senescence markers such as P16-INK4A and SS-galactosidase in adipose tissue and liver. Importantly, this decline in senescent cell burden corresponds with improved tissue insulin sensitivity, normalized lipid storage, and reduced SASP cytokine expression. In fat tissue, which becomes a major reservoir of senescent cells in aging, NMN has shown promise in restoring adipocyte function and mitochondrial activity. By facilitating autophagic turnover, NMN appears to limit the pro-inflammatory transformation of adipose progenitor cells and promote healthier adipocyte renewal. This effect is particularly important because senescent adipocytes and stromal cells impair lipid storage and endocrine function, driving ectopic fat accumulation in liver and muscle. The removal of these senescent cells reopens the door to metabolically active fat remodeling and improved energy partitioning, both of which are crucial to reversing age-related obesity. The liver also benefits significantly from NMN's support of autophagy-mediated senescent cell clearance. Hepatocytes are prone to DNA and mitochondrial damage over time, especially in the context of overnutrition and oxidative stress. Without effective autophagic and immune surveillance, senescent hepatocytes contribute to steatosis, fibrotic signaling, and systemic inflammation. NMN helps restore mitophagy and lysosomal capacity in aged liver tissue, reducing oxidative load and removing senescent cells that disrupt metabolic enzyme activity. In aged mice, these changes are linked to reduced liver triglyceride content, lower ALT levels, and improved hepatic insulin response. Skeletal muscle, although not a common site for classical senescence, does harbor dysfunctional satellite cells and inflammatory macrophages that contribute to metabolic decline in aging. NMN's activation of autophagy in muscle helps preserve mitochondrial networks, reduce ROS production, and clear senescent-like immune cells, supporting better insulin-stimulated glucose uptake and preventing sarcopenic obesity. Moreover, by restoring systemic NAD+ balance, NMN indirectly improves immune surveillance mechanisms such as macrophage phagocytic function and T-cell activation, further aiding in senescent cell clearance across tissues. Importantly, NMN's approach differs from senolytic drugs in that it does not forcibly eliminate senescent cells, but instead reactivates the body's natural maintenance machinery, reducing senescent cell formation and enhancing their removal over time. This gentler strategy may offer a safer and more sustainable way to manage cellular aging, particularly in older individuals or those with metabolic disorders who may be more vulnerable to the side effects of targeted senolytic agents. In summary, NMN's capacity to enhance autophagy offers a powerful tool not only for metabolic rejuvenation but also for gradual senescent cell clearance. By restoring NAD+ levels and activating key quality control pathways, NMN supports the body's ability to identify and remove dysfunctional cells that otherwise accumulate and drive obesity and metabolic dysfunction with age. This dual role, boosting metabolic efficiency while reducing inflammatory cell burden, suggests NMN could serve as a cornerstone therapy in combating both the biological and clinical manifestations of aging. As the science continues to evolve, NMN may represent a bridge between preventive anti-aging strategies and regenerative metabolic medicine.